Hi everyone, it's me again, Dr. GB, and as promised, I'm going to talk about three factors that led to the fast development of the COVID vaccine. And we're going to start with the first reason, which is technology. Okay, so the last record for vaccine development was 4 years, and that was in 1967 with a mumps vaccine. All right, so with the technology we had available in 1967, in the 1960s, it took us 4 years. I want you to take a minute and think back to how different life was in the 1960s versus now, specifically how different the technology was. At that point, we hadn't put people on the moon. We didn't have the internet. We didn't have email. We didn't have cell phones. All right? So there's some big changes between now and 1967, but there were parallel advances in technology that were made in the biotechnology sector. So in addition to having technologies that makes our lives of communication easier, we have tools that make biochemical research and understanding viruses a lot easier and a lot quicker, and we get better and more detailed information. All right, so I'm going to talk about two technologies in particular that were super important. All right, so in previous videos, we talked about how proteins are made from mRNA, which is copied from DNA. So to be able, or in some viruses actually, and the coronavirus is one of them, actually have double-stranded RNA as their genetic material instead of DNA. So fun fact. So to be able to develop a vaccine, we need to know the sequence of the genome, or what the individual building blocks are, or in what order they are, or know the recipe that the genetic material in the virus codes for. All right. So we were not able to determine the sequence of the genetic material within a virus until 1995. And we didn't know it for humans until 2003. Okay. It's 2021 now. 2003 wasn't that long ago. And so we cleared that milestone in the late 90s, early 2000s, but it really hasn't been until the last 10, 15 years that we have been able to determine DNA sequences or RNA sequences in an organism quickly through techniques called high throughput sequencing. And so the technology for DNA and RNA sequencing has gotten a lot better in the past 10, 15, even 5 years. And those advances have allowed scientists in Wuhan to determine the sequence of the coronavirus within 2 months of the first sample being taken from a patient in the hospital in 2019.